Okay, let's begin. So, hi everybody, thanks for joining me on a Sunday evening. All right, so my name is Hanjin. I'm actually the Sec 2 Science tutor at OVERMUGGED. All right, so for today, we are going to be covering all the Term 3 and Term 4 topics. So that will be electrical systems, digestive system, transport system, as well as human reproductive system. Now, if you're wondering, you're not going to go through Term 1, Term 2 topics, man. All right, it is already on YouTube. So you just go and find on the OVERMUGGED YouTube channel. We did one for mid-year exams earlier this year. Okay, so for today, we'll only be focusing on these 4 topics. Now, if you haven't already downloaded or printed out the materials for today, please do so using this QR code. If not, I think I linked the Google Drive link in the chat. So we will start now with the first topic of electrical systems. What do you need to know? Also, before this, right, just to let you guys know, try to stay to the end of this session because there will be a Kahoot game and whoever's on top will have a chance to win one of the upper secondary curated notes, okay, which is worth like $40 or something. So yes, try your best to stay for the whole session. If you can't, it's okay, just leave. You don't have to message me or tell me anything, all right? Okay, so electrical systems, what do you need to know? First of all, there are 3 main components, okay? So there's electrical current, potential difference, and resistance. Let's examine what is current. The definition of electric current is the flow of electrons in one direction in a circuit. Okay, so the analogy I like to use for all my students is think of a river. All right, in a river, you have the flow of water. Okay, that is exactly the same. We call that current, right? Oh, we say, oh, the current of the Kallang River very strong. All right, because we're referring to the flow of water. So in an electric circuit, it will be the flow of electrons. A common misconception, people will be like, oh, current means electrons. No, it is not the same thing. It is the flow of electrons. All right, now there's 2 models that we can use. One is called the conventional current model. And the second one is called the electron flow model. What's the difference between these 2? Conventional current model just says that, oh, okay, it flows from the positive end to the negative end of your battery, while electron flow says that it flows from the negative end to the positive end. Which one do we use in exams? If they do not specify, you always use conventional current, which means that when you're tracing your current in the circuit, always trace from the positive end to the negative end. All right, we always use the conventional current model. To measure current, we will use, amperes as our SI unit. And we always, draw our ammeter, which is a device to measure current in series. What does this mean? Let's say you have a circuit, right? And then you have a bulb. Okay, and they're like, oh, how do we measure the current of this circuit? We will draw our ammeter on the main wire. That is what it means to be drawn in series, which means when we are tracing our current, it will pass through that ammeter back to the power source. All right, so we know for sure, okay, I've drawn this in series, it is correct. So that is electrical current. Now we'll go to the second one, potential difference. What is it? Another word for it is voltage. All right, so if you see your paper, voltage, potential difference, they mean the exact same thing. What is the definition? It's a very long definition. All you have to remember for me is that it is the energy needed to move a charge across 2 points of an electrical component. You're like, what does that mean? Okay, I'll break it down further for you. Okay, so let me draw a very simple circuit for you. Same, just one bulb in this circuit. If I connect my voltmeter over here, essentially all I'm asking you, right, is that, okay, this is point A and this is point B. All I'm asking is, what is the energy needed to move a charge from point A to point B? All right, so all you have to think about when you are answering any sort of potential difference or voltage questions, right, is where is my point A and where is my point B? This means, right, if I draw my voltmeter like this, I'm asking you what is the voltage of the component between A and B. For this particular circuit, there's only one thing between A and B. It is this bulb. So whatever we measure, right, if the voltmeter says, oh, this is 5 volts, okay, it means that this bulb, okay, has a voltage of 5 volts. All right, so let's say I change the circuit now. Let's say I draw something like this. Okay, we have 2 light bulbs. All right, and I draw and I connect a voltmeter like this. Where's my point A? Where's my point B? Point A is here, point B is here. I'm asking you, all right, so what is the energy needed to move a charge from point A over here all the way to point B over here? And how many components does it pass through? It passes through these 2 bulbs. So I'm asking you, what is the voltage of these 2 bulbs? All right, okay, so all you have to do, right, is to figure out where is my point A and where is my point B. Okay, whatever is in between point A and point B, all right, is, okay, the voltage of whatever it is you're measuring. Which is why, all right, if you guys see in some questions, if you have any circuit, and then you have a voltmeter that's connected here. Okay, I draw a voltmeter connected here. All right, what are they asking us for? Okay, this is my point A, this is my point B. What is it between point A and point B? As you can see, it is your power source. So they're asking you, what is the total potential difference of this circuit? Okay, what is the voltage or what is the potential difference of the power source? Okay, so just take note of this. I know it's a bit confusing for some people. All right, so just really all you have to do is remember, okay? Figure out where is point A, where is point B, okay? And what is in between point A and B. Now, the last component, right, will be resistance. So that is a measure of opposition to current flow, which means, okay, resistance is always opposite to current. So when resistance goes up, current has to come down. When resistance goes down, current has to come up. So resistance and current, they have a inverse relationship. This is important because a lot of questions are asking like, oh, what happens if I vary the resistance? How would that affect current? So you have to know that there's an inverse relationship. When resistance goes up, current will go down. All right, so this is also what I've written here. The greater the resistance, the lower the current. And how do we measure current? We use ohms. Now, there's 2 types of resistors that we can use. One is a fixed resistor. It just looks like a box, okay, or a rectangular box, okay. And the other one is a variable resistor, which is the same thing, but with an arrow passing through, okay, in this direction. So you just need to know for fixed resistor, right, what is the function of a fixed resistor? It just adds resistance to the circuit. So all it does is it has resistance. Okay, but for variable resistor, it has a slightly different function. It varies the resistance. Okay, so the resistance can go higher, can go lower, and ultimately that is going to affect the current. So you guys must know, all right, what is the function of fixed as well as variable resistors. It is somewhere probably in your textbook. Okay, will it come in exams? Not very likely, but it still could. So it'll be good to know. Okay, some functions of fixed and variable resistors. Now we are going to explore series versus parallel circuit. So this is arguably one of the biggest parts of this topic. All right, so first of all, how do you identify if it's a series or parallel circuit? Okay, series circuit, right? If you trace the current from the power source all the way back, like from positive to negative end, you will pass through all the wires. Okay, so there's only one main wire. But for parallel circuit, when you trace it, okay, you will realize, hey, I didn't pass through all my wires. All right, there were branches. Okay, there was stuff that's outside of the main wire. So as long as we have branches, right, we consider it a parallel circuit. Okay, it doesn't matter 1 branch, 2 branch, 3 branch, 4 branch. Okay, as long as when you trace your current, right, you realize, hey, I couldn't trace everything in one go. Okay, it means it is a parallel circuit. Now, you have to be aware of these things in the table. So first of all, what happens when a bulb blows in a series versus parallel circuit? So for series, let's say this bulb blew. What's going to happen to the other bulb? All right, you need to remember that all other bulbs will be affected. okay, all other bulbs will blow, which means that no bulb will be lit. All right, so if one goes down, all of them will go down. Why? Because you see, you trace your current. In order for the bulb to light up, the current has to go back to the power source. But you see here, this one blew. This one was like totally faulty. So you took it out. Okay, there's a hole. All right, so you can see the current gets stuck here. It's like a roadblock, the construction on the highway. Okay, you can't go past this. So the current will never be able to reach back to the power source. All bulbs will be affected. No bulbs will be lit. That is for series circuit. How about parallel? Parallel, let's say I blow the first bulb. Okay, now one thing to understand about parallel circuits is that, okay, because there are branches, right? Okay, the current will split. What does it mean when the current split? Think about it. If I have a river, okay, this is your Kallang River. All right, now it can split into smaller lakes or smaller rivers. Okay, like going to different directions. So it's the same thing for a parallel circuit. It can split. So when you come back to this parallel circuit, if let's say the first bulb blew, if you trace the current to the first branch, you're like, hey, it got stuck. Okay, I couldn't manage to go back. It's okay. But can it go through the second branch? Yes. As you can see, there's still a pathway for the current to go all the way back to the power source, which means that this particular light bulb can remain lit. So what I'm trying to say is that in a parallel circuit, right, if one bulb blows in, okay, that particular branch, all right, the other bulbs in the other branches, all right, like this one, or if I add another branch here, or as many branches as I want, all the rest of the branches are unaffected. Okay, so the bulbs in the other loops will remain lit. Okay, now let's explore current. In a series circuit, right, for current, it is the same throughout, which means if I put an ammeter here and I measure current and it tells me, oh, this is 10 amps. Wherever I move this ammeter, okay, wherever I move it on the series circuit, it will give me 10 amps. So this represents total current. Take note, okay? If you have an ammeter on a series circuit, it will always represent total current. Okay? But for parallel circuit, current is shared between the loops. What does this mean? It means, okay, the total current of a parallel circuit is split between the different branches. So let's say first branch, if I put ammeter here, it gives me 10 amps. And then the second branch, if I put ammeter here, it gives me 5 amps. The total current in this circuit will be 15 amps. All right, it will be split between the branches. All right, so just take note of that. Then for voltage, it is just the opposite. So for voltage in a series circuit, total voltage is the sum of voltages, which means if I tell you this bulb here is 5 volts, this one here is 8 volts, the total voltage will be 13 volts. All right, so that means your battery has a voltage of 13. But for parallel, once again, it's the opposite. So we always start with what's in the power source. Let's say this battery is 6 volts. Okay, 6 volts will be supplied to each branch. So 6 volts is going to come here and 6 volts is going to come here. All right, so each of these light bulbs will have 6 volts each. Okay, it doesn't matter whether I have 2 branches or have 10,000 branches. Okay, as many branches as I want. Each branch will receive 6 volts. Okay, so that's why I say the total voltage flows through each branch. All right, now the very last one we're going to talk about will be resistance. So for series, is similar to potential difference, which means if the total resistance is a sum of individual resistances. So if here I have 5 ohms of resistance and here I have 5 ohms of resistance, my total resistance will be 10 ohms. Okay, but for parallel, it's slightly different. There is a formula to determine total resistance. Now, if you're a lecturer, I've never seen this in my life, okay? It means that your school does not expect you to know this. which means you can ignore what I say for the next 2 minutes. But if your school expects you to calculate resistance, then you must know this formula. Okay, it's a very important formula. Essentially, right, all you have to understand is that resistance in a parallel circuit, right, okay, is that the more branches you add, okay, the lower the total resistance. So it's an inverse relationship. For series, okay, the higher the number of like resistance you add, okay, the higher the total resistance. But for parallel circuit, it's opposite. The more you add, okay, the lower the total resistance. So I want to talk about how it relates to adding bulbs. So this is a very common question. Okay, they say, what happens if I add, okay, a light bulb in a series? Okay, if you add a light bulb in series, it becomes dimmer. Okay, but if you add a light bulb in parallel, it becomes brighter. Why? Because in series, okay, the more bulbs you add, the total resistance increases. Okay, remember what's one of the first things that we mentioned today? Okay, resistance is related to current. So resistance and current have an inverse relationship. So if total resistance increases, total current will decrease, okay, and your bulb becomes dimmer. All right, but for parallel, if you add more branches, total resistance decreases, so total current will increase and the bulb becomes brighter. Okay, so please take note of this. Now, for Ohm's law, once again, not all of your schools will be testing Ohm's law, but for those that are, you have to be very familiar with the V equals IR equation. Okay, so for anything, you have to be able to use V equals IR. All right, and use this, just have to move the things around. So for example, if you want current, okay, you just move, become V over R. Or if you want resistance, okay, that will just be V over I. All right, same for power. For those schools that are testing power, remember for me, the equation for power. Okay, power in watts will be electrical energy in joules over time in seconds. Your units are very important. Okay, most of your schools will be testing questions where they say like, oh, in minutes. So you have to remember the equation is in seconds. So you have to convert your values back to seconds to use the power equation. Okay, so we are nearing the end of this one. Okay, let's talk about wirings in a main plug. So any 3-pin plug in your house contains 3 types of wires. All right, you have your live, neutral, and earth wire. So what you need to know? You need to know, first of all, position. So which wire is which, okay, in both views, this one and the cut open one. What else? You need to remember for me, color, okay, the color of each wire, as well as current and potential difference. So in a live wire, right, okay, there is current as well as potential difference. Okay, in a neutral wire, remember the name is neutral, which means okay, it's like it doesn't really care, okay, it doesn't want to do anything. Okay, so got current but no potential difference. Then last one, okay, will be your, okay, this is the wrong one. This is your earth wire. Sorry. Your earth wire, okay, is just a safety precaution. It will have no current and no potential difference if, you know, your appliance is working fine. Okay, so for electrical dangers and precautions, all you have to do is memorize for me. Okay, so what are some of the important dangers? You have damaged wires, wet conditions, overloading, and short circuit. I don't think I have to really explain this. Okay, memorize it for your exams. Okay, you have to tell me what is the danger and what is the consequence. So you have to tell me damaged wires, okay, can result in an electric shock. Wet conditions, okay, what happens? Also a shock. Then for overloading and short circuit, it can cause a fire. All right, so link it to a consequence. Then for precautions, 3 main precautions. So first one, earth wiring. What's important? Remember, it's connected to the metal casing. Okay, always is connected to the metal casing. What does it do? It conducts the current to the earth. Okay, then for fuse, what is it? It's just a small metallic wire connected to the live wire. Okay, it's connected to the live wire because that is the one that is dangerous. Okay, so the fuse is connected to the live wire. What happens? If the current is too high, it will melt and break the circuit. And the last one, circuit breaker in your house also have. What does it do? If your current is too high, it will trip and break the circuit. Sorry, what is going on? Okay, yes, and it will break the circuit. Okay, can? So now I want you guys to take 5 minutes, right, to try this question. So this is mainly for people who have schools that are testing Ohm's law. So V equals IR. If your school is not testing this, you all can just take a 5-minute break. Okay, so for the benefit of those who need to learn Ohm's law, okay, can you all try this question and I'll go through in 5 minutes. Okay. All right, I'm going to go through the question now in the interest of time. So the figure below shows 3 resistors connected to a battery with a EMF of 10 volts. Okay, I don't know why it says 3 resistors. I only see 2. So part A, state the potential difference across the 2 ohm resistor. Now, before we can even solve anything, right, let's figure out, is this a series or a parallel circuit? How do we figure out? I told you, just trace the current. If when you trace, okay, back to the power source, if it passes through all the wires, it's a series. If it can't, it's a parallel. So if I'm tracing, okay, I'm like, oh crap. All right, as you can see, there's some parts of the wires that did not go through that tracing. So this is definitely a parallel circuit. So when we're answering this question, right, we should be referring to on top all of the things about parallel circuits. Okay, so over here, what's essentially going on is that when the current is coming out of the power source, at this point over here, it's going to branch, okay, into branch 1 and branch 2. Okay, it's going to join back together and come back to the power source. So for part A, state the potential difference across the 2 ohm resistor. Remember, it depends on your total voltage. Your total voltage is 10 volts, which means that 10 volts is going to be supplied to each branch. Okay, remember, in a parallel circuit, your total voltage is given to each and every branch. It doesn't matter if I have 50 branches. Every branch will get the same amount as the total, okay, your power source. So the answer for this is 10 volts. Now for part B, calculate the effective resistance between A, B. So you can see where is A, where is B? A is over here, B is over here. What did I mention about potential difference? All we're asking is what is in between point A and point B. So what's in between point A and point B? Okay, you have 2 components. You have the 4 ohm resistor as well as the 2 ohm resistor. All right, so basically this question is asking, and these are the only 2 components of this circuit. So this part B is asking you, right, what is the total resistance of this entire circuit? All right, what is the formula for resistance for parallel circuits? Okay, for part B, okay, 1 over total resistance equals to 1 over the resistance of branch 1, okay, which is 4, plus 1 over the resistance of branch 2, which is 2 ohms. All right, so that is going to give you, okay, 3 over 4. But remember, this is 1 over total resistance. So my total resistance is going to be the flipped version, which is 4 over 3 ohms. Okay, so answer is 4 over 3 ohms. All right, you can leave it as 4 over 3 or you can leave it as 1.33 to 3 significant figures. All right, then for part C, find the current, okay, flowing through point A. So you're like, okay, where is point A? All right, point A is over here, okay, on the left. So that represents, as you can see from the color coding, that represents total current. Okay, so if it's on the yellow portion, we're talking about total current before anything split. Okay, on the red is current of branch 1. The blue is current of branch 2. Okay, so at point A, we're still asking what is the total current of the circuit. How do we find it? We have to use Ohm's law. Okay, we have to use V equals to IR. So for part C, all right, let me shrink this. Okay, so for part C, right, first things first, write out your V equals IR. Okay, we are looking for current. So we want I equals to V over R. Do we have total voltage? Yes, our total voltage is the power source, 10 volts. Okay, 10. Do we have total resistance? Yes, we just found that from part B. Okay, which is 4 over 3. Okay, so 10 divided by 4 over 3, that is going to give me an answer of 7.5 amperes. Okay, so final answer 7.5 amperes. All right, I hope you guys were able to solve this question because this is a very standard Ohm's law electricity parallel circuit question. Okay, can? Very good. Now, we are going to go straight into the second topic of the day, digestive system. Now, first things first, we need to figure out what's the 3 main nutrients. What are the nutrients that we consume? Okay, everything in life can be broken down into carbs, proteins, and fats. What do you need to know? You need to know the function. So for carbs, what does it do for our body? It provides energy. Proteins, growth and repair of damaged tissues. Fats, insulation as well as energy. Important, memorize this. Okay, function of each of the nutrients. All right, next important thing, they will ask you, why do we need to digest? Why is it I can't just, have these carbs enter my cells directly, okay, and provide us with energy? Why did we bother digesting them into simpler molecules? Okay, so for that, right, you need to remember, okay, that, okay, the reason why we digest is because these nutrients are too big to pass through, okay, the pores of the cell membrane. Okay, ignore the bad handwriting. But yes, it is too big to pass through the pores of your cell membrane. Okay, so no matter how hard you try, okay, fats, proteins, carbs, they literally can't enter your cells. Okay, so they can't, okay, do their functions. All right, they have to be broken down into simpler, smaller molecules, okay, that can pass through the pores of your cell membrane, okay, in order to be used by your cells. Okay, so also remember for me, your answers, right, you have to include, okay, the reason, okay, or the rationale behind why we digest food. Okay, then, so for chemical digestion, okay, we always involve, okay, digestive enzymes. So you also need to remember some key characteristics of enzymes. For example, all enzymes in your body, okay, digestive enzymes or any other type of enzyme, all of them are made up of proteins. They speed up the rate of reactions. They are reused after a reaction, and they are specific. What is specificity of an enzyme? This means if this enzyme is only for breaking down carbohydrates, it will never be able to break down fats and proteins. All right, so for example, if you see over here, we have 3 classes of enzymes for 3 types of nutrients, right? We have carbs, proteins, and fats. So we have 3 classes of enzymes. We have carbohydrases, proteases, and lipases. Each of these classes of enzymes will digest one type of nutrient. So the first one is carbohydrates. It breaks down your large carbohydrate molecules into simple sugars. For example, breaking down starch into maltose. What is one example I want you guys to remember? Okay, is a name, amylase. So this is one type of carbohydrates, and amylase breaks down starch into maltose. So you see the word amylase later on when we are figuring out what happens in our organs. But this is quite important. So if you see the word amylase in your exams, remember it is a carbohydrates. It breaks down carbohydrates into simple sugars. Then next one, we have protease. Is in the name. What does it do? It breaks down proteins into amino acids. All right, so amino acids are the end products of protein digestion. The last one, we have lipase. It breaks down fats into 2 things, fatty acids and glycerol. All right, so this is the only one that breaks it down into 2 different things. Why? Because if you look at the structure of fats, okay, you can see it's literally made up of 2 different things, okay, fatty acid as well as glycerol. Yes, so yes, it's broken down into 2 separate things. Now, some of your exams, right, will ask, they will ask something like, oh, okay, they did an experiment, all right, and then the end product of digestion of that compound, like compound X, okay, is acidic. So they're asking, is it carbohydrate, protein, or fat digestion? And it's very simple. The answer will always be fat digestion. Why? Remember, fat is broken down into fatty acid and glycerol. What is the name of this? It literally says fatty acid. Okay, the name has the word acid, which means that fatty acid is a type of acid. It is acidic. All right, so when you break down fats into fatty acids and glycerol, okay, the end product will be acidic. The rest, okay, amino acids, glucose, etc., okay, are neutral. All right, so just take note, if in your exam you see it, you confirm know, oh, confirm is fat digestion. Okay, so what's the other main thing about this topic? You need to remember what are the key processes that happen in each organ in your digestive tract. All right, that is 5 key organs from the mouth to the large intestine. Okay, so for mouth, what exactly happens here? In the mouth, one of the key things that happen is chewing. What does chewing do? Increase surface area. All right, so that salivary enzymes, okay, can work more efficiently. So always link back to increased surface area. Remember, like when you're eating bread, okay, when you chew the bread, okay, it breaks down into smaller pieces of bread, okay, that have larger surface area. Okay, are there enzymes in the mouth? Yes, one specific enzyme. You have salivary amylase. So we just went through this. Amylase is a type of carbohydrates. So in the mouth, there is only carbohydrate digestion. Okay, so your fats, your proteins, nothing will be digested in your mouth. Only carbohydrates because of the presence of salivary amylase. All right. Now, the second organ is your esophagus. It is just a long tube that connects your mouth to your stomach. Okay, you see this tube over here? This tube is your esophagus. You spell OE or you spell E for your esophagus, doesn't matter. It's just one is British English, one is American English. All right, so esophagus. What is one key process that happens here? Something called peristalsis. It's just the contraction and relaxation of your muscles in the esophagus. All right, why is that important? Because it wants to push your food down. You might be thinking, oh, but gravity will do the job, right? If it's like falling down a slide. Yes, most of the time gravity helps. But imagine you take a Big Mac, okay, and then you lie on your bed. How is that Big Mac ball of food gonna go down your esophagus if you're lying flat down? All right, so you still need some muscles, okay, in your esophagus to help push it down, regardless of whether you're standing upright, you're lying flat down, or you're doing a handstand. Okay, can? Take note, no digestion. There is no digestion that takes place in the esophagus. Now, for stomach, what is important? We have gastric juice that is made up of hydrochloric acid and proteases. So the hydrochloric acid kills the bacteria in the food, and your proteases does what the protease does, okay? It breaks down your proteins into amino acids. So take note, your stomach is very acidic because of the presence of hydrochloric acid. All right. Yes, and it is also the reason why you don't get food poisoning after every meal, because the acid is killing all the bacteria from your food. So yes, what digestion happens in the stomach? Only protein. Why? Because gastric juice only contains proteases. So no carbohydrate, no fat digestion, only protein in the stomach. Now we are on to our fourth organ, that is our small intestine. It is the main site of digestion, which means, okay, main site means all 3 nutrients will be digested here. Carbs, proteins, fats, everything, zapalam together, okay, confirm will be digested in the small intestine. So there's a misconception. People think, oh, the stomach is where digestion happens. Not really, okay? The main site of digestion is the small intestine. There's a few juices that I need you to remember. The first one is called intestinal juice. Main function, okay, neutralization of acid. All right, when you go O-level, you all learn that, okay, actually intestinal juice also got other enzymes, but at your level, you just need to remember one of the main functions of intestinal juice is to neutralize the acid from the stomach. Okay, the bacteria kill already, everything kill in the stomach already. Okay, so when it comes to the small intestine, we don't need an acidic environment anymore. So we want to neutralize the acid. Then pancreatic juice contains your 3 key enzymes. Okay, so your amylase to digest carbohydrates, protease to digest fats, and lipase to digest your protease to digest proteins and lipase to digest fats. All right, so all 3 nutrients. And then last one, you have this thing called bile. Bile is not really a juice, okay? It is just something that helps in fat digestion. So we'll come back to that a little bit later, all right? But it helps fat digestion. It is not an enzyme. So take note, bile is not an enzyme. It does not digest the fat. It just helps, okay? And the small intestine is also the site of absorption. So at this point in small intestine, we've digested everything down to the end products of digestion. So it will be small enough to be taken up into the bloodstream and enter our cells. Okay. Last one is your large intestine. So here, once again, no digestion. Everything ends at the small intestine. Okay, whatever your body wants to digest, it will digest there. Okay, everything else, no more. Okay, it's going to come out of your body one way or another. All right, so what happens in the large intestine? It is a site of water reabsorption. So take note, in your large intestine, okay, there's no digestion, but your body wants to take back as much water as possible. Okay, water is precious to humans, so it will always want to take more water. All right, so yes, site of water reabsorption. Okay, so this table is very important. You have to be very, 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 very familiar with all the processes that happen at each organ. Okay, so at the top of your head, you must be able to tell me, okay, stomach is protein digestion, mouth is carbohydrate digestion, small intestine is all plus absorption, etc., etc. Okay, important. Now, last one, I want to go back to bile. So remember bile, okay, it aids fat digestion through a process called emulsification. Okay, if you're a lecturer, I've never heard of this, then ignore it again. Okay, but if you have, you're like, lecturer, I've heard of this, but I forgot. Then yes, please remember it. Okay, emulsification is the process where bile breaks up very big fat globules. So one big fat droplet into smaller fat droplets. So essentially, it is trying to increase surface area. All right, why is it important? Because remember, for fat digestion specifically, in which organs can it occur in? There's only one organ in which fat digestion occurs, and that is the small intestine. You see, it doesn't happen in the mouth, it doesn't happen in the esophagus, it doesn't happen in the stomach, it also doesn't happen in the large intestine. It only happens in the small intestine, which means it has so little time to really digest the fat. So it needs a helper, okay? It needs like a maid, okay? And that is bile. So it's helping to break up the fat globule into smaller fat droplets. All right, to increase surface area for more efficient digestion. Okay, so that wraps up for this topic. Now I'm going to give you guys 5 minutes to attempt this question, then I'll go through it again. Okay, I'm going to go through a question now. So sorry, we are running a little bit short of time. Okay, so part A, label structures A, B, C, D. So A, you can see is a tube that is connecting to the stomach. Okay, basically, there's only one thing that's really like a tube in the digestive tract. All right, that will be your esophagus. Now, part B, it is the one that I said is over here. Okay, what is that? That is your stomach. All right. Then for part C, okay, I realized part C is pointing to the stomach. Okay, then ignore that. Then for part B, what is that? That will be at the end of your entire digestive tract. All right, I didn't cover it, but I'm pretty sure you went through in your school. That will be your anus. All right, so part B, name the organ in which the absorption occurs. That is your small intestine. So recall, small intestine is the main site of digestion as well as the site of absorption. Now for part C, Sally eats a large amount of butter. Describe what happens as it undergoes chemical digestion in the various parts of her digestive system. To answer this question, first of all, you need to figure out what is butter, right? And butter is essentially made up of fats, lots and lots of fats. Okay, so this question is basically asking you, trace fat digestion as it goes through the digestive organs. Recall, where does fat digestion take place? Only one organ, all right, the small intestine. But you have to let the examiner know that you know, okay, that it can't take place in the other organs because lipase is not present. So you have to tell me that as it goes from the mouth to the esophagus, to the stomach, all right, as it passes through these organs, there is no fat digestion. So no butter digestion, basically, because there are no lipases present. So that's the first part of your answer. You have to tell me that, okay, actually, the butter, it goes through the mouth, the esophagus, the stomach, but no fat digestion because no lipases are present to digest it. Then it enters the small intestine, which is the site of fat digestion. Why? Because your pancreatic juice, okay, your pancreatic juice contains lipases to digest fats into fatty acids plus glycerol. Okay, so you tell me that in the small intestine, all right, there is fat digestion because, okay, there's lipase in pancreatic juice to digest the fats into fatty acids and glycerol. All right, so the end products of fat digestion, your fatty acids and glycerol, will be absorbed into your bloodstream. Okay, if you want to go one step further, if this is a 4-mark, 5-mark question, you also talk about bile. Okay, why? Remember, bile aids in fat digestion through emulsification of fats. All right, but let's say this is a 2 to 3-mark question. Okay, then whatever I wrote over here is sufficient. Okay, can? Very good. So we are going to go into our third topic of today, and that is transport system. What's important? You need to, first of all, memorize diffusion and osmosis. Must memorize the definition. 3 marks in your exam. You don't take your classmate will take it. Okay? So remember, diffusion, net movement of molecules from a region of higher concentration to lower concentration. Think about it. Okay, when someone sprays deodorant after PE, okay, after a while the whole class can smell it. Why? Because the deodorant particles diffused across the classroom. For osmosis, it is only water molecules. Okay? No other types of molecules. Only water molecules from a region of higher water, okay, ignore this, higher water potential to region of lower water potential. Okay, don't use the term water concentration. Sorry, I don't know why it was written like that. Okay, say higher water potential to lower water potential. Now, take note, osmosis must have a partially permeable membrane. Not, oh, can. I'm talking about must. Okay, there must be a partially permeable membrane for osmosis to take place. All right, whether that be a whisking tubing or whether that be a cell membrane, all right, doesn't matter as long as there is a partially permeable membrane. So next thing is, how do plant and animal cells act in different types of solutions? So there's 3 types of solutions: hypertonic, isotonic, hypotonic. All right. Just remember, okay, so for hypertonic solutions, essentially, right, it is a solution with, okay, very very low water potential. Okay, so for example, it's concentrated sucrose solution, concentrated syrup solution, concentrated Ribena. Okay, very low water potential. What's going to happen when you put a plant cell inside? Water molecules are moving out of the cell, moving from the cell, okay, out of the cell into the concentrated solution. What's going to happen? The plant cell is going to become plasmolysed. Then for hypotonic, it's just the opposite, okay? It's very high water potential. For example, distilled water, okay? Very dilute sucrose solution, dilute Milo, whatever, dilute anything now, okay? Hypotonic, it has high water potential. So water is going to move from solution into cell. So water is going to move into the cell. What's going to happen? It's going to start swelling and it becomes turgid. Then last one, isotonic, is same water potential. So no net movement, no change. Okay, so for animal cells, it's exactly the same thing. All right, exactly the same. Hypertonic means low water potential. So water moves out of the animal cell. Okay, what's going to happen? It's going to look like a raisin. Okay, we say that the cell becomes crenated. Okay, the terms are important now. We don't say plasmolysed for animal cells. We say crenated. Then for hypotonic, it's high water potential solution. So water molecules move from solution into cell. Now, take note, in animal cell, the cell will burst and die. All right, it does not become turgid. It dies. All right, why? Because they do not have a cell wall. Okay, animal cells don't have a cell wall. So when too much water enters the cell, it's just going to burst. Think about it. If you put too much water in a water balloon, eventually it's also going to burst. All right, but for plant cells, because there's a presence of a cell wall, okay, the cell wall is like the walls of your house. Okay, so you pump a lot of water in, your house won't explode, okay, from too much water. But for animal cells, no cell wall, so it will burst and die. Then last one, isotonic, same, no net movement, no change. What's next? Components of blood. So remember, in blood, there's 4 main components: red blood cell, white blood cell, platelet, plasma. Okay, you need to know how they look like. Yes. Okay, so red blood cells always have a donut-looking shape. Okay, white blood cells have an irregular shape. You will see, as in you will know it when you see it, and we'll have a question on that later. Okay, then platelets is very, very small, and plasma is just the fluid. Okay, so plasma is not any cell. All right, 4 components of blood. What do you need to know? You need to know the function. So red blood cell, transport oxygen, return carbon dioxide. Main function is the transport of oxygen. You also need to remember the 3 key features of red blood cells. No nucleus, contain hemoglobin, and biconcave. So for no nucleus and biconcave, this one and this one, okay, 1 and 3, it is to increase the surface area. All right, why? Because it wants to pack more oxygen molecules. Okay, if not, it has to make so many trips. But no, we're like, no, I want to pack as much oxygen as I can. So I want no nucleus and I want a biconcave shape to increase surface area. Then the second one is contain hemoglobin. What is that? It is just the substance that binds to oxygen. Okay, it's like sticky, so it binds to the oxygen. All right, make sure that the oxygen doesn't fall off randomly. Okay, then for white blood cell, immune function, what does it do? Fight bacteria, produce antibodies. Just memorize it. Platelets form blood clots. Okay, stop bleeding. And last one, plasma, transport nutrients and waste substances. So take note, transport of oxygen is red blood cell. Transport of everything else, okay, your nutrients like your glucose, amino acids, whatever, okay, is plasma. Okay, waste products like urea, also plasma. Okay, so red blood cell is transport oxygen. All right, so memorize it for me. Now for blood vessels, you need to know there's 3 types of vessels: capillaries, veins, and arteries. The big one will be artery and vein. In between those 2, you have capillaries. Okay, you can see how thin and how small capillaries are. All right, so what do you need to know? You need to know the features of each type of blood vessel. So for example, all arteries carry blood away from the heart, but veins carry blood towards the heart. How are capillaries? It connects arteries and veins, okay? And the key function of capillaries is exchange of substances. So all your oxygen and everything, the waste products, right, go through capillaries to be exchanged between blood and body cells. All right, then you also need to know for arteries, the blood that's flowing through it, very high pressure. How does it withstand that kind of high pressure? It needs a thick muscle layer. So they are interconnected. All right, then for veins, okay, we don't need such a high pressure. Okay, low pressure, so we need a thinner muscle layer. We don't need it to be so thick. All right, then last one is lumen. You're like, sure, what is lumen? Lumen is just the hole in the center that allows the blood to flow through. So if you see a cross section, okay, this is the muscle layer outside, right? This hole in the center, okay, whatever's inside, this refers to the lumen. All right, so lumen size is obviously just opposite to your muscle layer size. If you have a thick muscle layer, like I drew over here, you have a very thin lumen. But if you have a very thin muscle layer, so this is your muscle layer, you will have a very wide lumen. All right, so this is narrow, and this is wide lumen. All right, so just remember for me, artery, the blood need to be very high pressure, a thick muscle layer, which also means it has narrow lumen. Then for veins, don't need so high pressure. All right, so thin muscle layer and wide lumen. Okay, last one, plant transport system. What do you need to know? Please, you'll cover this in primary 6. Okay, so 2 types of transport vessels, xylem and phloem. Xylem transports water and mineral salts. Phloem transports sugars from photosynthesis. Now, take note, specifically what sugar? Sucrose. Take note, it transports sucrose. So you're like, sure, but I thought glucose is the one that is produced in photosynthesis. Yes, glucose is produced in photosynthesis. It will be converted into sucrose for transport. All right, you're like, sure, why? I don't know, ask the plant. Okay, so sucrose is the one that is transported. Okay, then direction. Xylem, obviously, because you take it out from the roots. So the water and mineral salts move from the roots to the rest of the plant. Then for phloem, because photosynthesis happens in the leaves, all right, all these sucrose is transported from the leaves to the rest of the plant. Why is it important? You need to know which is which when there's a cross section. 2 main cross sections, root and stem. For the one that's most common is stem cross section. All right, the one on the right. One way is just to memorize. Okay, so the xylem is always the one in the center, and the phloem is the one outside. Same as the root cross section. So xylem is the one in the center, and phloem is the one outside. Now, if you're my student, you will know that I have a brilliant analogy for this. Okay, which is that water is so precious to all living things. All right, we can only live 3 days without water, but we can live 1 week without food. So water is 100% more important to us. Now, if something is important to you, do you keep it near to you or do you keep it far away from your center? You obviously keep it near to your center, right? Like if you have your phone, you keep your phone in your hand, in your pocket. You won't leave your phone at the edge of a kopitiam table. All right, so it's the same. If it's precious, we keep it near to our center. So your xylem is the one carrying water. Water is precious. So the xylem vessel is always nearer to the center. You can see xylem vessel is nearer to the center in both the stem cross-section and the root cross-section. Okay, so be very aware of that. So yes, now we are going to try this question. Same now, okay? Take 5 minutes to try it, then we'll go through the question together. Okay, I'm going to go through the question now. And once again, please stop fighting in the chat, okay? Or I'll be kicking you out of the Zoom. Now, for this question, so we have 5 discs of fresh potato, all right, immersed in sugar solutions of different concentrations. Then obviously there's some changes in the mass. So either it reduced in mass or increased in mass. And then there's a graph to show the differences of each potato strip. So part A, name the process that causes the change in the mass of the potato discs. The answer will always be osmosis. So if you ever have any question in exams, okay, potato strip lah, sweet potato strip lah, carrot strip, whatever it is, okay, put in sugar solution, just remember the answer is always osmosis. All right, it will be the movement of water molecules. So part A, answer osmosis. Now for part B, arrange the potato discs in ascending order of concentration of the sugar solution. So from the least concentrated solution to the most concentrated solution. How do we answer a question like this? We need to think to ourselves, what does it mean to put this strip in a, let's say, a very dilute solution? All right, remember, we just went through this hypertonic, hypotonic, isotonic solutions. If it is a dilute solution, okay, does it have high or low water potential? It has high water potential, which means that the water molecules are going to move from solution to cell. All right, the water is going to move into the cell, into the potato disc. So water enters, all right, the potato disc. If it is a concentrated solution, it has low water potential, which means, all right, the solution has low water potential, which means that the water molecules are going to move from cell into the solution. It's going to move out of the cell into the solution. So water leaves the cell. All right, so this is the cell. Dilute solution means water enters the cell. Concentrated solution means water leaves the cell. So if water enters a cell, what do you think is going to happen to the mass? Mass is going to increase. For concentrated, if water leaves the cell, what do you think is going to happen to the mass? Mass is going to decrease. All right, so as you can see, if there is an increase in mass of the potato strip, it has to have been put in a dilute solution. All right, so that all the water from the dilute solution enters the cell. Then now you're asking, how do I know how do I know how diluted it is or how concentrated it is? So the steeper the concentration gradient, all right, the more water molecules will move across. Think about it. Why do molecules even bother moving, whether it's diffusion or osmosis? You are trying to reach equilibrium, trying to reach equal on both sides. So if one, wrong one. If one has 100 to 0, how many molecules must move across to make it equal? Okay, 50 molecules have to move across to make it 50-50. But if the difference between two solutions is, let's say, only 10 and 0, how many molecules must move across to make it equal? Only 5, okay, to make it 5-5. So the more concentrated it is, okay, the more movement, or the more dilute it is, the more movement. So the correct answer for part B should be C, D, B, E, A. All right, so potato strip C was put in the most dilute solution. So the most amount of water entered, it had the greatest increase in length. All right. Similarly for A, right, it was put in the most concentrated solution. So the most water left the cell. So there was the greatest decrease in the mass. All right, so for part C, explain what happened in potato disc C. So what caused such a great, increase in mass of the potato strip? All right, so to answer this question, we will always talk about water potential and osmosis. So you have to first of all tell me that the water potential of the cell set in potato disc C, I just put C, okay, water potential in the cell set of potato disc C is lower than the sugar solution, lower than the water potential of the sugar solution. So water potential is lower than the sugar solution. So what's going to happen? All right, water molecules are going to move from where to where? move from, okay, sugar solution into the cell set through what? Always remember, osmosis has to take place through a partially permeable membrane. So you have to tell me through the partially permeable cell membrane, okay, via osmosis. All right, via osmosis. So basically, even if your English is very broken, it doesn't matter because the teacher is going to mark it according to keywords. Okay, so you can tell me osmosis at the start, at the sentence, of the end of the sentence, whatever. All right, so basically water molecules are moving from sugar solution into cell set through the partially permeable membrane via the process of osmosis. Okay, I will answer questions at the end of the session, okay, because I'm afraid we're going to overrun the time a lot. All right, then you tell me that, therefore, the potato disc C will increase in mass. Okay, or you can tell me increase in mass the greatest. All right, so take note for bio, always about keywords. Okay, now last topic, reproductive system. Okay, what's important? I'm sorry, I have to rush through some stuff, okay? You must know the location of the different organs. If your school is not testing this, you can just shut off your brain or you can leave the call, okay? But if you want to stay for the Kahoot, then you just chill. All right, so this topic, you need to know the location of the organ. So where's the ovary, where's the fallopian tube, where's the uterus, cervix, and vagina. Then you need to know the function of each of these organs. Okay, very important. The function is important. Same for male reproductive system. You need to know where these organs are located and what is the main function of each of these organs. Okay, also, each of these are female and male reproductive system. There's two views, front view, side view. You must know both views. Okay. Puberty. So what happens during puberty? What are you guys going through right now? Essentially in puberty, your sex organs will mature and you can produce your sex cells. So gametes is just another word for your sex cells. What are your sex cells? You have your sperm as well as your ova, your egg. Okay, so once you go through puberty, your body will start being able to produce and release these sex cells. And then you also go through a series of physical changes. So for boys, what exactly is going through? Your sperms will be produced, okay, deepening of your voice, facial and body hair development, as well as marked muscle development. All right, for girls, what's the different physical changes? Okay, you can start to release mature eggs. Okay, you start menstruation, your breasts will begin to develop and your hips will widen. Now for both, across both genders, you have growth spurt and you develop pubic and underarm hair. So yes, remember for me, what are the physical changes? We're going to go into menstrual cycle. This, okay, take note, menstrual cycle is not the same as menstruation. So menstrual cycle refers to the whole cycle, like it's a continuous process, okay? And part of your menstrual cycle is menstruation. So menstruation takes place in day 1 to 5. All right, so if a girl says, oh, I'm on her period, she's referring to menstruation, which is the discharge of your uterus lining, okay, through the vagina. So you're discharging blood and dead tissue through the vagina. That is menstruation. Okay, that's day 1 to 5. Now, from day 6 onwards, the sex hormone estrogen is going to repair and thicken that lining. Remember that all of that lining has been shed out of the body in day 1 to 5. So there's nothing left. So day 6 onwards, the body is going to start repairing and rebuilding that lining. All right. Now, for day 11 to 16, we call that the fertile period. So this is the period that leads to the highest chance of pregnancy. Okay. So take note, different schools will have different dates. So some schools will say 11 to 16, some will say 10 to 15, some will say 10 to 16. Follow your school notes, okay? Don't follow mine. I mean, if your school doesn't specify, then follow mine. But if your school has a different range of the fertile period, please follow your school's notes, okay? And specifically on day 14, that is the day that ovulation happens, which is when a mature egg or ovum is released from one ovary. Okay, can? Then after that, day 17 onwards, we don't need to grow the lining anymore. It has already been repaired, has grown to a sufficient thickness. So progesterone, another sex hormone, is going to maintain the lining. Okay, why do my notes have so many errors? Okay, so the lining maintains the thickness. Okay, so progesterone primarily maintains the thickness of the lining. All right, but estrogen over here repairs and thickens the lining. All right, so slight difference. Okay, but there are two main key hormones, okay, estrogen and progesterone. All right, now last one, birth control methods. Same, memorize it for me. Okay, so you need to know how each of these birth control methods work. Okay, so for example, the key ones, condom, you need to know there's a male and a female version. So the male one, okay, is just a rubber tube, okay, that you put over the penis that prevents the deposition of sperm. All right, so it's like a plastic bag. It prevents any sperm from leaving and entering the female. Okay, now for female, the female condom is called diaphragm. Okay, it also prevents sperm from entering. So it's like an umbrella that stops it. Okay, then we have intrauterine device. What does it do? Okay, it is fitted, okay, into the wall of the uterus, okay, so that it prevents the fertilized egg from implanting itself in the wall. Okay, so it physically blocks, okay, the uterus lining. So even when fertilization takes place, you form your zygote, right? Your zygote can't implant itself in the uterus wall. Okay, it's blocked by the IUD. And last one is contraceptive pill. It just stops ovulation. Okay, it alters like the chemicals and the hormones in your body. All right, then permanent, important. Permanent means permanent. Okay, no going back. So for males, it's called vasectomy. You cut and tie sperm ducts. Then for females, tubal ligation. You cut and tie fallopian tubes. Okay, very last one, STIs. 3 main STIs. Remember for me what they're caused by. Gonorrhea, syphilis is bacterial. HIV is a virus. Okay, then you need to remember the key symptoms. So for gonorrhea, burning sensation when you pee and pus being discharged. For syphilis, you have sores and non-itchy rashes. Okay, and last one, HIV, the virus causes AIDS. Okay, why? Because it kills off all the white blood cells in your body. Okay, so that is a key thing to take note. Now, in the interest of time, I'm just going to do this question on the spot with you guys. Okay, so label structures P, Q, and R. This is the front view of the female reproductive system. P, you can see it is pointing to this like bean-shaped thing, right? This is your ovary. Okay, then Q over here, it is the narrow opening that is called the cervix. Then R, okay, is referring to this canal that is your vagina. Okay, so must remember where each of these organs are. Now, state one function of structure R. So there's a few there's a few functions that you can talk about for the vagina. So one will be, okay, is the birth canal. All right, it's the birth canal. You can also say that it's the site of sperm deposition. So this is where sperms are deposited. All right, then for part C, so very long, but essentially you're asking, all right, what's happening here? They say infertile, okay, so this woman carries out in vitro fertilization. Then where should this fertilized egg be implanted back in the woman? So where does the zygote get implanted? It is in the uterus lining. So we just need to X out here. Okay, left, right doesn't matter. Okay. Okay. Now, we are done with this. So I'm going to open a Kahoot right now. Okay, what is going on, guys? Hold on, let me. Okay, sorry, give me 10 seconds, guys. Okay, I've made one person the host. Okay, I'm going to message this person. Can this person please help me regulate the chat? If you need to ban people, just go and ban. Or if even better, just like stop the chat altogether. So like, yes. Please, can you please stop with these racist comments? Like, it's damn sickening. Okay, you guys are what, already 14. For those who are still remaining, I'm so sorry for what's happening in the chat. We are going to proceed with the Kahoot now. Then for the admin, please just ban the chat if you can.
Yeah, sure. You know, you can win like $30 notes. No, like she said it's worth $30. Yes, it is $30.
Okay, sorry guys, I'm a bit technically challenged. Hold on, I'm setting up the Kahoot. Okay, I got it. Oh no, oh my gosh. Okay, wait, can the host give me sharing abilities? Oh no, I'm sorry. Wait, can the host just, okay. Sorry, yeah. Okay, can you see, please? Can you see my screen?
No.
Okay, I'm so sorry. Can you see it now? Okay, great. I'll start in 15 seconds. So whoever haven't joined, please join now.
What are you?
Okay, I am starting now. Okay, guys, essentially, right, their question is just asking what happens if I increase the resistance, right? They say what happens if I increase the resistance of the rheostat. All right, remember, resistance is inversely related to current. So if you increase the resistance, you will decrease the current. So the ammeter reading decreases. Okay, great. All right, so this one is just memorize only, all right? So remember, fuse is always connected to the live wire. All right, so remember, what's one of the key processes that happen in the mouth? All right, chewing. What does chewing do? It increases the surface area. All right, it has nothing to do with proteins and fats. Okay, so by the time the food enters the esophagus, it has a larger surface area. Okay, so basically they're asking which parts of the digestive system did the meat patty digestion occur? Meat patty is what? Is protein lah, okay? So protein where? Stomach, small intestine. All right, so the answer is 1 and 3. So which component of blood is involved in transfer of oxygen? That would be your red blood cells. All right, so red blood cells always looks like a donut. It has a biconcave shape. That will be structure Y. Oh my gosh, oh my god, I said this question wrongly. It's D. I'm so sorry to the 71 of y'all. And to the 20 of y'all is wrong. Okay, is option D. I'm so sorry, I saw it wrongly. This is my fault. I'm sorry, guys. You still have 2 more questions to redeem yourselves. Okay, so a vasectomy is a permanent method of birth control where you cut and tie the sperm ducts. All right, so all you have to do is you need to just figure out which of the structures is the sperm ducts, and it will be the tube that's connected to your testes. All right, so that will be option A. Okay, last question, let's go. Great. Okay, you guys know that because day 14 is the date of ovulation. So that's when the mature ovum is released. Okay, but even if you totally didn't think about that, all right, what's the fertile period? Day 11 to 16. Okay, there's only one option that lies within the fertile period. That is day 14 here. Yay. So let's take a look at the podium. I'm so sorry, guys. I feel like so many of y'all should have gotten 8 out of 8. This is my fault. Yay. Okay, very good. To whoever this sure person is, right? Can you please PM me on Telegram, okay, at HANJUNN. All right. Just screenshot the page that says that you are sure. All right, so that I know that it's you. Okay. Yes, and then I'll contact you and you can get your curated notes. Okay, so for everyone else, thank you so much for joining me today. I'm really very, 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 very sorry about what happened in the chat. I didn't know that this was going on when I was like doing my talking. And to those who participated in whatever was going on, okay, shame on you. You should be very ashamed that you are talking all these racist things. Okay, this should never have happened. All right, everyone else, I hope it was an enjoyable and productive session. Please study hard for your end of year exams. Okay? Yes, that's all for today. Bye.